Namaskar and welcome in special conversation with Doordarshan. We are having Dr. EICHLER from University of Washington, who is a leading genetics scientist in the world, and he has been nominated for Times 100 Influential People in 2022, and he has been researching in DNA and genetics area for many decades. We'll be talking to him. Dr. EVAN, welcome to Doordarshan, welcome to India.
My pleasure.
Yes. First question I would like to ask you that in layman's terms, if you can explain your body of work, that what you have done in like genetics, genome mapping, and all your understanding about evolution of genomic, human genome evolution. So please tell us about that.
Sure. I've been interested in genetics for almost 30 years. Was part of the actual initial Human Genome Project, which was in the late 1990s and early 2000s. And at that point, I was studying genetic causes of developmental disability, autism, and epilepsy. And my particular focus is to find the regions of the human genome that are changing very rapidly, with a very simple idea that these areas that change very rapidly will contribute disproportionately to both disease and evolution. And so I've built my research career on applying genomic technologies to do two things. One is to identify genetic causes of developmental disability in children with unexplained developmental disability, epilepsy, congenital heart defects. The second thing, which is a little bit different, is this hypothesis that we have that those same regions are actually helping to make us uniquely human. So they are contributing to genetic differences that help specify who we are as a species and distinguishing us from other non-human apes, such as chimpanzee and gorilla. So my whole life has been dedicated to that research. And much of the impact has been in terms of actually solving genetic cases of unexplained disease. But because the regions that I study typically change very, very rapidly, these are regions that have not been really well characterized until recently.
Okay, so there are very unstable regions. So there may be non-coding regions as well in the DNA. So how do you find out and how do you get the hold of that region by looking into it, or what interests you to go there? Because in 2001 around that we got human genome mapped. And we could really read the exomes which were used to make proteins for humankind. So why there is a need to go into that region?
Well, in 2001, the map that we had, which was very complete to a point, it was the first human reference genome, it missed many of these regions. We knew they were there, and this goes back to some old history, but we knew they were there because there were two different approaches to sequence the human genome. And we combined those approaches and we realized that these pieces of information were missing. So we predicted back then in 2002 that those regions would be unstable. And we tested specifically children with developmental disability to see if they were unstable in them and not in the general population. And when we did this, we discovered about a dozen or 24 new genetic causes of a developmental disability. But then we started to wonder, those same regions that were making us have children that have autism, developmental delay, and epilepsy, why do these unstable regions exist at all? So we went back and we looked, and we found in those regions were actually genes. And those genes are part of the instability, but at the same time, those genes are important in terms of it's helping to build a bigger brain. So increasing the number of connections, the number of neurons, the number of synapses, and so on. So that kind of led to the whole hypothesis that we need to understand both the stable and the unstable regions if we want to understand what makes us human and if we want to understand what causes us to have disease. So they're both, I don't know if you have this term in India, the yin and the yang.
Yes.
It's a balance. And in our genome in particular, we are wired for both disease, but also the things that make us uniquely human are embedded in those regions.
So good and evil both are the part of same human being.
It is, and you can't have one without the other, right? Because you could say, well, if these things cause disease, why don't we get rid of those regions, right? If you could. But the part of the matter is that they actually carry the things that distinguish us from all other species on the planet. So I became fascinated with that, and then what has happened in the last few years is the ability to use new sequencing technologies, we call them long-read sequencing technologies, to finally completely sequence both the stable and the unstable portions of the genome.
So this is a very interesting thing that you are saying. That how that some portion of genome which really no not helping human being, they are producing disease and everything, but still they are part of our evolution, they are part of our survival, they are making us a better as a species and as a race. So how do you explain this thing?
Because those regions make us more adaptive.
Okay.
So other species don't have them and they still live. But there are things that make humans more adaptive to certain things, like social interactions, expanding culture, expanding language, all these things. And part of that is related to the development of the brain. And so our interest is actually correlating those instabilities. We know there's a cost, but the benefit is in terms of making us, in many ways, uniquely human. So that's, in order to understand these regions, you have to be able to access them. So, go back 20 years, those regions weren't there in the human genome. And now in 2022, we finished the one human genome, and now we're sequencing hundreds of human genomes because we think there is more diversity among us, and it's in these regions of the genome, and they're going to play an important role in both disease as well as the things that make us unique as a species.
So what we are going to do with this knowledge?
Well, one thing that we're doing, and we've already started, we've done this over the last 15 years, it started with these big deletions and duplications that we found in these unstable regions. And then technology evolved and allowed us to find specific changes in genes. We've been able to go back to the families. And when you say a child is born with intellectual disability autism, you talk to the families and they feel like they're isolated in the world. No, they don't know anybody who has exactly the same type of feature as their child. And what we've been able to do is, because we're screening now tens of thousands of cases, we've been able to identify the 5th and the 6th and the 7th genetic cause. And what we've been able to do now is bring those families together and say, oh, your child has the same genetic cause of developmental disability. The power for that for families has been tremendous. So some of my clinical colleagues say, well, what is the point? Because you do this, but we don't have a treatment for those children right now. Well, I say not yet. But there's something much more valuable than just actually having an immediate treatment. It's basically helping people understand why their child has intellectual disability, developmental delay, epilepsy. But more importantly, once you've identified the 10th and the 11th and the 12th case, those families actually now connect based on the same genetic cause of disease. And so what I have found from personal experiences of dealing with those families, when they reach out to me, I don't seek them, they come back to us and they say, thank you, because I don't feel alone anymore. I feel like I have a family that's outside of my family because we have shared experiences. And to me, and I'm not a clinician, but it is so powerful when you have, 50-year-old mothers telling a 25-year-old mother, this is what works, this doesn't work. And that information is invaluable to the families. A feeling of no longer isolated, but also practical things about raising a child with developmental delay until we have a cure. And the other thing, which I think is going to be something I'll never see, but when the treatments are developed, and they will be developed, those families that have a genetic cause that are type 1, type 2, type 17, they will be the first in line to actually receive the treatments because they know the genetic causes and they are the most informed families on the planet.
Okay. So when we talk about the scientific journey, I would also like to ask you about your personal emotional journey, a passionate journey that you had over the last 20, 25 or 30 odd years. So how do you see yourself in this journey as being a person, being an emotional person?
I guess for me, part of the motivation for doing genetics is the human side of it. And that has been the thing that's always why I didn't go and work in mice genetics or Drosophila genetics, because I wanted to make a difference in people's lives. I was personally motivated because I have a niece with autism. Okay. And like many autistic cases, we haven't solved her yet. And I believe there is a genetic cause that remains to be found. And that's why I continue to actually work on developing, that's a personal motivation, but developing these methods that we're doing now where we'll understand all the genetic variation that exists in any genome. And to me, that's why I've been motivated to go after really the parts that people have left behind, because I believe part of the answer lies in those parts.
Today we are living in time where we are living with CRISPR technologies. We have Google DeepMind who does AI prediction for protein foldings, and lots of big data we are having. As you might have also contributed to some of the very big huge database as well, which might be used by all these technologies. So why don't you think that these technologies are not able to solve yet with this much of data? The problem that we are having, why we are not able to find the drugs, why we are not able to find those receptors, those what is lacking? Are we something behind? Is there something still missing? As you might have felt in 2001 or 2 that these parts are missing, and you try to fill that gap. So is this the same thing right now in 2024 as well?
Yeah, I think so. I mean, I think part of the problem is that we have gapped information. So what I mean by that is when people study children with all kinds of disorders, or even adults, people have preconceived notions of what parts are important and functional. And so they study those regions very well, but they don't understand it in the context of everything. So the other thing related to that, so basically being able to finish genomes completely means that we're able to understand, or at least discover, not understand, discover genetic variation completely. So that's why it's important to keep the eye on the goal, which is complete discovery. And most technologies over the last few years have not been complete. But now the technologies have advanced that we can be complete. So that's point number one. Point number two, which I think is actually really important, and I put it squarely on geneticists. In human genetics in particular, there's a monogenic focus. And what do I mean by that? That means once I have discovered the gene that I know is already important in some cases, I've solved the case. So I'm done, okay? But we know with people with the same genetic cause of disease, this is more common than not, they can be extremely different in terms of their phenotypes. Some can be severely affected, some can be mild, and so on. So I think the problem is that we're too centered on a single gene model, that we have to be thinking about understanding all the variants that make who you are, not just the ones that we think we understand. But I think it's the synthesis of all that variation that's important. So the reason I think we still haven't solved a lot of genetics is, number one, we didn't comprehensively characterize genetic variation. But that problem is solved, in my opinion, now. But number two, we're still stuck on this idea of one gene, one mutation causes one disease. And that's true for some diseases, but I don't think it's true for most of human genetic diseases. So it's not until we consider all the variation, maybe this is where AI comes in. Maybe this is where some of this technology can be leveraged, right? To actually start to synthesize all the variation that makes you unique as a person, but also contributes you to unique risks of disease.
So you mean to say that we really yet don't know how genes interact with each other?
Exactly. So we understand genes, we understand the basics. If you think of it as a textbook, The Human, a novel, a beautiful novel, with a few typos. We are understanding the letters. We're just beginning to understand some of the words, but we haven't even begun to put together sentences yet. And that requires understanding how genes encode proteins that interact to make every living cell in your body, right? And understanding that if I have a mutation in 10 locations, that's the sum of those mutations that will give me a risk to coronary heart disease, right? Not just one gene. And I think that's part of the problem with our field. We need to be comprehensive in terms of our discovery, which we haven't been till now. And now we need to synergize the variation to explain outcome.
So do we really think, like it's a hypothesis, like do genes talk to each other?
All the time.
All the time.
They talk through each other through the proteins that they encode, right? So almost no gene lives in isolation. Right? It actually produces a protein that interacts as part of a bigger complex. That complex interacts with another set of complexes, can modify, enhance, change. And so it's constantly dynamic. It's constantly moving, changing in terms of its interactions. But those pathways are highly predictive, right? But they take time to figure out. And so this is critical information. Is actually, people are working on this. But the idea of actually linking that back to the mutations comes back to the comprehensive discovery again.
what role can India, with its diverse population, diverse genetic population, can play and contribute in the research work, and what kind of collaboration and opportunities that you see in the Indian support?
Well, I think the diversity of India is actually a strength, I think, and the numbers of Indians is also a strength. The reason for that is that for many diseases, each is individually rare, but collectively common. Okay, so autism is the one I know the best. 500 at least different genetic causes. All told, that's 1% of the population that has it, but each one is their own type. Okay, so in order to understand a disease, you really have to have extremely large population base. And now India and China are unique in this regard, right? With respect to the diversity, I think it also is a benefit because the diversity actually allows you to really look at changes in allele frequency so that some populations will have higher and lower, and you can use that information to predict the disease prevalence that is different in different populations. But having said that, I think it's critical that India, like every other nation, identify a subset of genomes from diverse peoples from all across the subcontinent and sequence them very well to understand the natural pattern of variation that sits in this subcontinent. I think it's critical, and it's missing right now. I mean, in almost all of the analysis that we have done, there's some contribution from India, but it's a small piece. It doesn't represent its proportion in the world, right? And I think it would be great if India could, I mean, I know there are lots of issues that a country has to deal with, right? It's not, genetics is not the only one. But this is a real opportunity because I do think the future will be complete genetic information for all of your citizens, right?
Okay, correct. So when we look at the larger picture, still in this time, what parts of the science, what parts of genomic science are missing, which are not able to, which are not enabling us to see a complete picture?
Oh, there's lots of missing pieces, right? I mean, we talked about the missing information in terms of genetic variation, which we think we're on the cusp of solving. So that's good. Understanding the diversity of humans is critical. That will be solved eventually at some point. But actually, truly, the function of all the individual genes and how they interact. There's lots of people working on this problem, but it's not easily solved because you have to actually look at many different tissues, many different cell types, know all the interactions. You have to test those interactions and so on. So that will take probably generations to really build that information out. And then there are changes above that. So there are changes that occur in the cells that go to make your arm or your hand or your eye or whatever. The somatic changes, how often do they occur? What is the rates of those mutations? Those are contributing to your overall health. I mean, most cancer is the result of the somatic mutational process. And while we have glimpses of all of these things, by no means do we have a complete encyclopedic knowledge. So I think that should be a high priority. But then the other flip side of that, and I don't blame anybody for doing this, is that we should be working on treatments. And we should be developing treatments engineered to the genetic causes and not just treating symptoms, which is so much of what medicine is treatment of symptoms. And I think what we need to do is understand root causes and try to ameliorate, maybe not immediately cure, but to mitigate the consequences of this and do it in a molecularly informed way. So we call that in the States, I don't know if you have this term, personalized medicine.
Yeah, precision medicine.
Or precision medicine. Yes. And I think that is more than a pipe dream. I think it is becoming a reality. But it starts by, like anything else, you build the foundation first, right? You build up, you understand your variants, your frequencies, you characterize the Indian population, and then you go forward and you find the disease-causing variation, and then you go forward and you develop treatments. And some of the treatments may be very specific, right, to specific diseases and specific populations in the end. But of course, drug companies don't like this because they want to sell a drug that everybody will take. That's how you make your most money. If you have a drug that will only affect 400 families in the world, that's not a good investment of resources, right?
Correct.
So it's a complicated world, right?
Yes.
We need the drug companies to be developing the drugs. But the idea of precision medicine is one that isn't, from a financial perspective, maybe the most appealing.
So, when you are talking about personalized medicines, let me talk to you about Indian system of traditional drugs that we used to make. It's Ayurveda. So, if you have fever and I have fever, so a doctor over here, a person who is practicing Ayurveda, will make a different medicine for you according to your nature, and he will prescribe a different medicine for me according to my nature. Since the empirical data that we have for this modern medicine is around 200, 300, 400 years old, but traditional system around the world, whether it is Siddha, whether it is Ayurveda, whether it is Greek, Unani, they are almost around 2,500 years old. So, don't this traditional system of knowledge is helping you to see the body and the problems that we are seeing in a different way, if you have encountered some insights from ancient understanding?
Well, there's no question. I mean, it's the same in China as well, right? They have what they call Eastern medicine and Western medicine clinics that are set up for both. And I spent some time there. And I believe that in some herbal remedies, there are actually some essence. There's some treatments that are actually very effective, right? And people don't quite understand, and pharmaceutical companies have made models of trying to actually understand, what is going on at the molecular level. So it's interesting that you equate this to, the personal nature versus precision medicine. I think the difference I would make is that when I assess your nature, you assess mine. It's a subjective of it, no matter how good you are.
Nature of a body.
Nature of a body. But there's a subjective aspect to it. The power of genetics, it's objective. So this is what I think should be guiding, right? So this is why I think there's such precision medicine doesn't have to involve genetics, but it often does, because the precision comes from an objective function, which is understanding the genetic cause, and then saying, okay, now I know this is the gene that's mutated. Let's design a molecular therapy that's going to be specific to your family, or maybe people like you and your family, based on a genetic cause. So that to me is the difference, right? I have great arguments with my mother, who's also a herbal medicine person, right, about this. But I don't discount it. Right?
Yeah, I'm not telling that these both are I just want both are against each other, but I just want whether that knowledge is helping you to understand the modern problems, or is that something that the Western world is not looking at, the knowledge that East has produced for many of years. So sometimes what we feel is that as you say, Western medicine has very much specific approach. They look into the bits and pieces. They don't look in the totality of the knowledge that we have as a human species.
I think it's a little bit of a fair assessment. There's a paternalistic nature of Western medicine in general. And it's not just, it has to do with Eastern medicines. It has to do with how patients used to be treated in themselves, right? In my grandparents' generation, I think they almost would consider the physician like the god, like what he said that was spoken from heaven, right? But it's not so, right? I think one of the things that good doctors now versus bad doctors, the difference is doctors that listen to their patients. It's that simple. And listening to your patients, the patients have something to say nowadays, much more than they did in the past. And I think the same when we think about our genetic information. All that information should be taken into account, right? And I often say that the best treatments will be ones that involve three groups of people: the researchers, the clinicians, and the families. And there should be a three-way street where everybody communicates as equals, not as someone who's superior. Because I can tell you, I've learned more from families sometimes than any paper or textbook about essentially a new genetic disease that we've discovered, right? The insights that they have, because they live with them, that to me is tremendous, and families should be listened to, right? Which I think maybe that's where you're getting at with the Eastern medicines, because it is that rapport that's pretty important, right? That exists.
Correct. Do you have any thoughts regarding the consciousness or the way? It's a different topic, but just I would like to understand, because I think you dwell much into the philosophical part of the genetics as well. So definitely the genes are communicating. Definitely the body is working. It's perfectly working in the way every body is perfectly working. But from where does the life comes?
Consciousness. That's a great
Yes,
question.
from where does the life comes? The communication they are deciphering as the way Ivan is or the way I am. So how does the gene express the consciousness
So I mean, to
of the life?
be fair, I think that there is a part of who you are is not just genetic, right? A big part of who you are is who raised you, your experiences, your culture. All of this is important, right? Yeah. So part of who I am and who you are, we can't disentangle that, right, from that. But one thing I also will tell you, and I think it's self-obvious, humans breed true, which means that no matter where you are born, right, a human's going to be a human, right, in terms of their capacities. So when I come back to that, I think that there must be something wired genetically in us, right, to develop. And that may be connected to increasing the number of synapses, right, increasing the number of neurons. And some of these dynamic regions may also be contributing to the variability that we see in terms of that consciousness. And so there is actually a gene family that we're studying right now that is extremely high copy in humans and low in most other species. And it's very variable between humans. And we're very interested. It plays a role in the developing brain. We're just beginning to actually characterize that gene family. But we think it's going to be important in maybe some aspect of development of our brain, right? And the plasticity of our brain, which is, I think, a fascinating thing. I mean, you realize evolutionarily, right? As a species, we are very young.
Correct.
Right? So when we think about, if you believe the fossil record and the humans that existed before us, the archaics, right, the ancients, and it's only been, as a species, we separated from the other apes about 5 to 6 million years ago, which sounds like a lot, but in the history of this world, it is a drop in the bucket. And then as a species, our brain developed and expanded in only the last million years of our evolution. And culture, the way we recognize culture, in itself is only tens of thousands of years old. So that's amazing when you think about it, right? How do all of those changes, every human that's born is born with a bigger brain and a delay in, essentially the spine formation, the plasticity, all these things. There's things that make us uniquely human, but every human does that. And that all has to have been encoded in our DNA very recently. So this is, to me, the biggest question, and the one that I care about since I was a student, right? And that comes back to that yin and the yang, right? I don't think when people study disease without putting it in an evolutionary context, I think they're foolish. And I think the same way about evolution in humans. Not thinking about the phenotypic consequence in terms of disease and studying evolution is foolish, because they're totally intertwined, right?
Correct. So when does life begin? When
Oh, that I cannot pretend to understand that.
So when there is sperm, then there is egg. So they both interact. They both talk to each other when there is a. So at genetic level, when does life begin?
That is a great question, and it's beyond my pay grade. To be honest, I do not know the answer to that. But I think the potential for humans exists even in the very first conceptus, when the egg and the sperm come together, right? The potential for a human is now established, right? Of course, there's a whole bunch of steps in between, right? You go to the 32 cell stage, the 64 cell stage, gastrulation, neurulation. There's a whole process, right? But each of those is refining you more and more. But there's also, which people don't appreciate a lot, is there's a lot of stochasticism as well. And so that's why even identical twins aren't identical, right? Yeah. Because there's just slight changes in the whole process, which can have some significant impact in terms of how people are who they are, right? But at what moment does it begin? The potential is there from the single cell stage. The potential is there. I would say that. But yeah, it's a great question. That I don't know.
Thank you, Ivan. Thank you so much for talking so deeply and talking about the genetic justice that you're talking. And I think that our viewers will be really enlightened by this talk and the way we see the disease and the healthy mind and healthy body. I think it will add some perception to that.
Well, thank you. Thank you for your questions.
Thank you.
Appreciate it. Bye-bye.